Today we're going to start in on the chapter in your book about the large biological molecules that make all of these biological processes work. And we're going to break this into a couple of parts and work on this over 2 class days. So we're going to start off this part of the chapter on carbohydrates and lipids. So first of all, let's just think a little bit about these large biological molecules in general. It may not surprise you to know that all living things are made up of just 4 classes of large biological molecules. Those of us who are used to thinking in terms of our dietary needs are very familiar with the idea of carbohydrates, lipids, and proteins, and the need to balance these appropriately in your diet for optimal health. In spite of a recent study which, and I swear I'm not making this up, found that 80% of Americans would be in favor of mandatory labeling of foods that contain DNA, not making that up. Most of us who actually consume other organisms, which is basically everybody, don't tend to concern ourselves with the amount of nucleic acid we get in our diet, but it is there as part of these biological tissues that we consume. So these are the 4 major categories of biological molecules: carbohydrates, lipids, proteins, and nucleic acids. And when we talk about macromolecules, we're talking about large and complex molecules that are put together from a large number of basic parts, and these can be absolutely enormous strings of these carbon backbones that we talked about last time, and can weigh up to hundreds of thousands of daltons in molecular weight. And as we would expect from this principle of emergent properties, large biomolecules have unique properties that arise from the arrangement of these atoms in these large molecules and how the molecules themselves are arranged in space. This idea of folding of these large biological molecules. We'll think a lot more about that when we get to proteins in the next section of this chapter. So the majority of these large complex macromolecules are found in the form of polymers, which are constructed from individual units called monomers. So a polymer is a long molecule consisting of many similar building blocks, and those building blocks, those repeating units, are called monomers. So of our 4 major categories of macromolecules, carbohydrates, proteins, and nucleic acids are polymers. This doesn't mean that lipids are not put together from other subcomponents, but we don't have this pattern of repeating building block units in the form of these monomers repeating over and over and over again. So how do we get these polymers? How do we construct them? What are the connectors in our Lego set? So first of all, enzymes are hugely important. These are a category of proteins that work as catalysts. Catalysts in general speed up chemical reactions without actually participating in the reaction, so the enzyme is the same before and after the reaction, but it helps to facilitate that chemical reaction. So enzymes are very important to this process of making or breaking polymers. So when you put together monomers to form a polymer, we call this a dehydration reaction. So what we mean by this is that between these 2 monomers, they're going to come together in a way that forms a water molecule that is removed from the growing polymer and allows these 2 monomers to attach. So it's going to involve the loss of a water molecule from the 2 monomers that are joining. A little bit more detail on that in just a moment. And the exact reverse happens when these polymers break down into their components. So polymers are disassembled into monomers by a process called hydrolysis, and that's going to take a water molecule from the environment and attach parts of it to the 2 breaking apart ends of the polymer to remove water from the environment and add the atoms that made up the water molecule to form the 2 monomers again. It's exactly the reverse. So let's take a look at a video that illustrates this. So here we have 2 monomers. Each of them have hydroxyl groups as part of them. And what we're going to see once this animation gets to this point is there's going to be an enzyme that comes in right here that holds these 2 close together And the hydroxyls are going to remove two hydrogens and an oxygen and then form these bonds using oxygen between the two components. When it comes to breaking these down, enzymes are going to be used again to bring these together, and a water molecule is going to be used to add two hydrogens and an oxygen to those two monomers to take the water from the environment, and we call this hydrolysis. So now that this is still, we can see again, so from one of these, the process of dehydration is going to take one of these hydrogens and both the hydrogen and the oxygen from the other one and connect the carbon skeleton from this one to that oxygen, releasing this hydrogen, and this oxygen, which come together to form water. And then the reverse of that happens with the hydrolysis reaction that breaks the polymer apart. So carbohydrates fulfill several roles in organisms, and we're going to talk about them individually. So they act as fuel. So carbohydrates are sugars or macromolecules that store and release energy. They're building materials, so they provide rigid structure to cells. And also they are involved with cell recognition and identity. So we'll talk about all of these different roles of carbohydrates as we talk about the different kinds of carbohydrates that contribute to each of these functions. So carbohydrates are going to include monosaccharides, which are one sugar monomers. These are the individual units. Keep in mind that a single sugar can include any number of carbons in the skeleton. It's not necessarily one carbon, it's one sugar, and so that's going to be the repeating unit that comes together to form these polymers, which can involve a few sugars, in which case we call them oligosaccharides, or a lot of sugars, we call them polysaccharides. Don't worry about the difference between oligosaccharides and polysaccharides for the purposes of this course. Any chain of sugars we're going to refer to as a polysaccharide. So some more of the characteristics of carbohydrates. They're going to have the general molecular formula of CH2O and some number of repeats of that. So it's going to be some multiple of one carbon, two hydrogens, and one oxygen. So carbo refers to the carbon, hydrate refers to the water, so C and an H2O. And N can vary from 3 to over 1,000. So we can have very, very long polymers of these carbohydrate compounds. Carbohydrates contain a carbonyl group, so this C double bonded to an oxygen, a CO bond. It's going to have hydroxyl groups, so we see many of these hydroxyl groups bonded to the carbon chain, and many carbon-hydrogen bonds. So most of these carbons also have a hydrogen attached to them. Since these carbonyl groups and the hydroxyl groups are polar, carbohydrates are hydrophilic, and we know that sugar dissolves quite readily in water, so that tells us that these are primarily polar hydrophilic molecules. So what are some of the characteristics of monosaccharides? How do they differ from each other? So monosaccharide monomers are simple sugars. We call these simple sugars as compared to complex carbohydrates that are going to be composed of sugar polymers. And they're going to structurally vary in 4 primary ways. So one is the location of the carbonyl group. So it can be here at the end, and we call those aldoses, or it can be connected to one of the carbons within the carbon chain, and we call those ketoses, or ketone sugars. Second, they can vary in the number of carbon atoms present. We can see here examples of trioses, pentoses, and hexoses, both aldose versions and ketose versions of all of these, that gives these carbohydrates fairly high diversity of structure. They can vary in the spatial arrangement of their atoms, so there can be different arrangements of the hydroxyl groups. We'll see a lot more about that as we go along. And they can come in either linear and ring forms. So typically in aqueous solution, which is how you would find them in an organism, it's going to be in water in an aqueous solution. So we generally think of these sugars as occurring in rings for the purposes of biology. So let's take a look at how these ring shapes form from the linear form. So here we have just the chain of carbons with their different functional groups attached. We can number these carbons so that we can keep track of them. So 1, 2, 3, 4, 5, and then 6. And when the ring forms, the number 1 carbon comes in close proximity to the number 5 carbon. So we can see it sort of bent around like this. This hydroxyl group from the number 5 carbon is going to interact with this carbonyl group from the number 1 carbon. The oxygen from the hydroxyl group is going to form a bond with the carbon from the carbonyl group, and this hydrogen is going to get transferred over to this oxygen to form another hydroxyl group. So what we end up with here is an oxygen between the number 1 and the number 5 carbon, and then the number 1 carbon now has a hydrogen, and instead of a carbonyl group, it's got a hydroxyl group. So this is how these rings form. Same molecular formula, different properties, obviously, in a ring form than in a chain form. And these can be arranged in different configurations. You can either have the hydroxyl group pointing in the same direction as the hydroxyl group on the number 2 carbon, and we call this the alpha configuration, or it can be pointing upwards, whereas the hydroxyl group on the carbon number 2 is pointing downwards, so it's far away from that other hydroxyl group, and we call this the beta configuration. So because these are going to interact with their neighbors differently depending on how far away they are, these are going to have different properties. So now let's think about the structure of polysaccharides. How do polysaccharides form? So these complex carbohydrates are, of course, polymers of these monosaccharide monomers. The simplest case is 2 sugars linked together to form a disaccharide. And this happens when a condensation reaction occurs. So remember, this is going to kick out a water molecule as these 2 simple sugars link together. And this reaction is going to occur between 2 hydroxyl groups. A covalent bond called a glycosidic linkage forms. This is called a glycosidic linkage, when 2 sugar monomers link together to form a longer carbohydrate chain. And then, of course, this can be reversed. These linkages can be broken by hydrolysis using water to separate those 2 sugar monomers. So a couple of examples of this happening. Dehydration reaction for maltose, we've got 2 glucose monomers, simple sugars with 6 carbons. The hydroxyl groups are going to interact, the hydroxyl groups from 2 of these. The whole hydroxyl will be removed from one of them. Just the hydrogen will be removed from the other one to form water. And then this oxygen is going to come and bond with the carbon ring. As you see here, the oxygen linking these 2 monomer rings together in this disaccharide called maltose. This was what we call a 1,4 glycosidic linkage, because carbon 1 is going to make a link with the carbon 4 on the other ring, but it doesn't necessarily have to be that way. So this is an example of creating sucrose from glucose and fructose. So you don't have to have the exact same 2 sugars as your monomers. You could have, in this case, this is a 6 carbon sugar linking with a 5 carbon ring. And again, we've got the 2 hydroxyl groups that are combining to boot out that water molecule. And in this case, what we have is a 1,2 glycosidic linkage, the carbon number 1 from the glucose making a linkage with carbon number 2 from the fructose ring to form sucrose. So now let's start to think about the function of these carbohydrates. One of the primary functions is energy storage, and the major energy storage carbohydrate that's used by plants is called starch. So starch is composed of alpha glucose monomers, and it tends to form a helix. And this can be in either of 2 forms. One is called amylose, and this is an unbranched starch with only alpha 1,4 glycosidic linkages. Or it can be in the form of amylopectin, which has branches in it, so that means that carbon, at some point in a carbon ring, there will be 2 other carbon rings attached to it to form these branch structures. In that case, you're going to have some alpha 1,6 glycosidic linkages to create those branches. And in the case of amylopectin, we see branches about once every 30 monomers. And here's a figure from your book showing the structure of these. Starch is the very simplest version, just a single helix, no branching of these glucose monomers repeating over and over and over again. Amylopectin, again, you occasionally get a branch coming off from that number 6 carbon on one of these rings, and still going to be helical, but you're going to have these branches coming off. In animals, the primary storage carbohydrate is known as glycogen. Glycogen is stored in the liver and also in muscle cells to be able to deploy that glycogen very quickly when the muscles need it for energy. And this glycogen can be broken down into glucose monomers when energy is required. So glycogen is highly branched. It's an alpha glucose polymer, and it's nearly identical to starch, aside from this high level of branching. In the case of glycogen, we see branches in about 1 out of every 10 monomers. So you can see lots of branches coming off from this illustration of a glycogen molecule, but basically still just a polymer of glucose. So now let's think about one of the other functions of carbohydrates, and that's its use as a structural material to give some rigidity and shape to cells. So in plants, that structural carbohydrate is primarily cellulose. So this is a structural polymer, and it forms a protective layer around plant cells called the cell wall. So every plant cell is surrounded by this rigid cell wall. And it is made of beta-glucose monomers. So again, this is that other isomer of glucose, joined by beta-1,4-glycosidic linkages. So what this means is that every other glucose is flipped with respect to the one before it. So you can think of them as being kind of upside down. So instead of generating a helix, it comes in the form of a simple linear molecule. Because of both this flipped structure, which we'll see in more detail in the next slide, and also because of this linear structure, that's going to permit hydrogen bonding to form between these strands, which is going to make this a very nice, firm, rigid mesh of these fibers to give that solid structural material. The polysaccharide cellulose, major component of the tough wall of plant cells, as we just said. Like starch, cellulose is a polymer of glucose, so both of these are just repeats of glucose, but the glycosidic linkages differ between these two. And the difference is based on the 2 different ring forms for glucose, alpha and beta. So first, let's just take a look at the general differences between these alpha and beta type linkages. So we're looking now at 2 examples, one of alpha glucose binding to alpha glucose or linking with alpha glucose to form maltose. We looked at this before, and here we see that the 2 hydroxyl groups are both on the same side of the carbon ring. So when we do this dehydration reaction, we get the oxygen pointing down in the same direction as the hydroxyl group on the next carbon. And we call this an alpha glycosidic linkage. In the case of the beta glycosidic linkage, in this case we're looking at beta galactose and beta glucose, which are isomers of each other. They have the same chemical formula, but one of them has the hydroxyl group on one side of the carbon ring, the other one has the hydroxyl group on the other side of the carbon ring. And so this is going to form a very different structure when we link these 2 together, removing water. So now this oxygen is pointing up with respect to the plane of the rings, and this is a beta glycosidic linkage. So the result of this is that one glucose is flipped with respect to the other one. So here we have this carbon with its hydroxyl group pointing down below the plane of the rings. This one's pointing up, whereas in the case of maltose with the alpha glycosidic linkages, they're both on the same side of the carbon ring. Again, that's going to change the properties. So more specifically, in the case of starch and cellulose, what we see with these alpha glycosidic linkages is the hydroxyl groups are all on the same side of the plane created by these carbon rings. And in the case of cellulose, they're flipped on every other on every other carbon ring. So here we have the hydroxyl group pointing down, pointing up, pointing down, pointing up, and same with these carbon and hydroxyl side chains. And so what this means is that when these line up next to each other, they can form these hydrogen bonds with the next fiber over. So these are very different kinds of linkages between starch and cellulose. The enzymes that digest starch by hydrolyzing alpha linkages can't hydrolyze beta linkages in cellulose. So in fact, it turns out that there are no animals that produce enzymes that actually break down cellulose. So in general, when we eat cellulose, we just don't digest it at all. It passes right through, and we call this insoluble fiber. Very good for colon cleansing, but we don't actually get any of the energy. We don't break those bonds or break down those polysaccharides in any way. Animals who do get the actual energy and break down those cellulose fibers use microbes that actually do produce the enzymes to digest cellulose, and they have symbiotic relationships with them so that the microbes break down the cellulose, and then the herbivore, and this is herbivores from cows to termites, have these symbiotic microbes that break down the cellulose for them. So termites actually can't digest wood by themselves. They need to have their symbiotic microbes to break down the cellulose. Another structural material that we find in animals and fungi is called chitin. So chitin is a structural polymer. Its monomer is a substance called N-acetylglucosamine. Don't worry about memorizing that. And its structure is similar to cellulose in having those beta 1,4-glycosidic linkages that flips every other sugar. And so again, it's going to form those hydrogen bonds between linear strands and be another good structural meshwork that's going to be fairly stable and give good structure. So this is part of the exoskeleton in insects and crustaceans. They use chitin, and fungi such as mushrooms also use chitin in their cells to produce that structural rigidity. A final structural carbohydrate that I just want you to be aware of is found in bacterial cell walls. So bacteria need to have structural support to maintain its cellular shape. And this carbohydrate is called peptidoglycan. It's got long backbones of alternating monosaccharides, just as we've seen, joined by beta glycosidic linkages. But instead of hydrogen bonds between these strands, what we see in the case of peptidoglycans is short amino acid chains that form peptide bonds, which we'll learn more about when we get into proteins. These are the kinds of bonds that amino acids make together to form proteins. And they're going to form these peptide bonds between the adjacent strands, again forming a stable mesh network between these different, these long strands of carbohydrate. And then a final function that I want you to be aware of for carbohydrates is identifying cells. So cells use carbohydrates as kind of name tags that they attach to proteins on their cell membranes. And so they display information on the outsides of their cells. And these can be glycoproteins, so carbohydrates attached to proteins, or they can be glycolipids. They can be carbohydrates attached directly to the lipids. And these glycoproteins and glycolipids are key molecules in cell-cell recognition, so identifying cells as self or non-self. And also they're important in signaling between cells, so communication between nearby cells is done by these short carbohydrate chains attached to the cell membranes. So now let's change gears a little bit and think about lipids. So the main concept here is that lipids are a diverse group of hydrophobic molecules. So lipids are the one class of large biological molecules that does not include true polymers. So they can be very large, they're composed of different subunits, but they're not repeating subunits the way polymers are repeating subunits of monomers. And the main unifying feature, the defining feature of lipids, is that they mix poorly, if at all, with water. They're hydrophobic. So the reason they're hydrophobic is that they consist mostly of hydrocarbons. So remember that these are nonpolar molecules. All of these bonds between the hydrogens and the carbons are nonpolar bonds, so they're not going to be attracted in any way to polar molecules. So among this diverse category of lipids, there are going to be three that we're going to really think about that are very biologically important, and those are fats, phospholipids, and steroids. And we'll take a look at each of those in turn right now. So what is a fat? Fats are constructed from two types of smaller molecules, glycerol and fatty acids. So what is glycerol? Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. So here's our 3-carbon chain with a hydroxyl group attached to each of the carbons. So we start with glycerol. And then we add fatty acids. So a fatty acid consists of a carboxyl group attached to a long hydrocarbon. So here we have our hydroxyl group, COOH at the end. And remember, these carboxyl groups are very good for making links with other molecules. This hydrogen from the glycerol is going to combine with this hydroxyl group from the carboxyl functional group, kick out a water molecule, and make this bond between the glycerol and the fatty acid. So here we see this. The hydroxyl group's been kicked out along with the hydrogen from this hydroxyl group, and we've got an oxygen linking the carbons from the glycerol to the carbon from the fatty acid. And this is going to happen on all 3 carbons in that glycerol molecule to form what's called a triglycerol or triacylglycerol. So these fatty acids that are attaching can be of any length, and they don't all have to be the same on all 3 of these carbons in the glycerol. So this gives a lot of diversity to the number of different kinds of fat molecules that can be made. And these can also differ in bond saturation, and that's going to affect the structural properties of the hydrocarbons in these 3 tails in the fat molecule. So what do we mean by saturation? So saturated hydrocarbon chains consist of only single bonds between the carbons. So this is going to have the maximum number of hydrogen atoms attached, and it's going to form a very regular molecule. So remember, when each of these carbons is making its maximum number of 4 bonds, it's going to take on a tetrahedral shape. So in the space filling model, we see a nice straight, regular chain of hydrocarbon. Unsaturated hydrocarbon chains have 1 or more double bonds in the hydrocarbon chain. So hydrogen atoms are removed to make a double bond, so there's not going to be as many hydrogens attached. And what this is going to do is going to form a kink in the chain. So remember, when there's a double bond, a double bonded carbon, it's going to form a planar shape, and it's going to disrupt this regular chain of the hydrocarbon, and it's going to look something like this. There's going to be this planar region that's going to cause a kink in this chain. So when you have these unsaturated hydrocarbon chains, when they all group together, they're not going to pack as tightly together. And so what we find is that these unsaturated fats tend to be liquid at room temperature because they're not going to pack as closely together as these nice regular straight hydrocarbons. Saturated fats are going to tend to be solid at room temperature because of the way that they're able to pack closely together, because they lack these kinks that are going to create space between the molecules. So of course, one of the primary functions of lipids is energy storage. We talked a little bit about energy storage in terms of carbohydrates, and we're going to come back to that now just to compare carbohydrates and lipids as storage molecules for chemical energy. So we know that carbohydrates also store chemical energy. And they do this during the process of photosynthesis, where plants harvest energy from the sunlight and store it in the bonds of carbohydrates. So they're going to take carbon dioxide, the carbon from carbon dioxide, and use it to construct these carbohydrates in a reaction that looks something like this. But why not just use the carbon dioxide? Carbon dioxide has carbon in it. It's got some energy stored in it. What we find is that carbohydrates have much more energy than carbon dioxide. Why? Because in these carbon-oxygen bonds, these are polar covalent bonds, the electrons are going to be held more tightly to that oxygen in this polar covalent bond, and they're going to have relatively low potential energy. In contrast, in these carbon-hydrogen and carbon-carbon bonds, these are nonpolar bonds where the electrons are shared more equally, and they're going to have higher potential energy. So if we extrapolate this to the case with fatty acids have even more energy because they have even more carbon-carbon and carbon-hydrogen bonds. If we compare the structure of carbon dioxide to a carbohydrate to a fatty acid, we see no carbon-carbon or carbon-hydrogen bonds, just these highly polar carbon-oxygen bonds in carbon dioxide. In a carbohydrate, remember we've got a carbonyl group and a whole bunch of hydroxyl groups that are very polar, and a lot of carbon-carbon bonds and a lot of carbon-hydrogen bonds. But this entire tail in a fatty acid is all hydrocarbon. It's all bonds between carbons and carbons or carbons and hydrogens. There's a lot of energy stored in these fatty acids, which is why when we're really storing up energy to sort of save for later use, we're going to do that in the form of fats because they really store a lot of energy. The next category of lipids I want to talk about is phospholipids. So phospholipids consist of a glycerol again. So here is our 3-carbon glycerol chain. But instead of having fatty acids attached to all 3 of the carbons, one of the carbons is going to be attached to a phosphate group, and that phosphate group is going to be in turn bonded to a highly polar molecule. So we see here choline, and it's got a positive charge interacting with that negative charge on the phosphate group. So what does that mean? That means that this part of our phospholipid is highly polar. We've got completely charged molecules interacting here. Very, very polar. We've got lots of carbon-oxygen bonds in this region. And then we have these 2 fatty acids that are attached just the way they would be in a fat molecule. And this area is completely nonpolar. Remember, these are just hydrocarbons down here. So we've got a charged or polar head, which is hydrophilic, and a nonpolar tail, which is hydrophobic. In these hydrocarbon chains, these are going to be fatty acids. In bacteria and eukaryotes, it's going to be a little bit different in this one kingdom of life, archaea. Isoprenoids are another category of hydrocarbon chain that you don't need to worry too much about what exactly that is. And the primary role of these phospholipids is to form cell membranes. So let's take a look at how they do that. So phospholipids are what we call amphipathic, and that's just what we just described, having a hydrophobic and a hydrophilic region. So we can diagram this in kind of a cartoony way, putting a hydrophilic head and these 2 hydrophobic tails to symbolize a phospholipid. So when these amphipathic lipids are placed in water, the hydrophilic heads are going to interact with the water. So these are going to be highly attracted to those polar water molecules and to each other, since they're also polar. So they're going to cluster together, and they're going to want to be exposed to water. They're going to get where the water is. The water is also going to be sort of scrambling to interact with these polar head regions, which is going to exclude these tails. So the hydrophobic tails are going to interact with each other just by virtue of the fact that they're being excluded by all of these polar regions, and they're going to tend to, says they're going to get away from the water. It would be more correct to say that the water is getting away from them because it wants to be with the other polar molecules. It tends to be with the other polar molecules. It's attracted to the other polar molecules. And so because of this exclusion of the hydrophobic tails from the hydrophilic regions, they're going to spontaneously form lipid bilayers. So in experiments where researchers want to have an isolated cell membrane by itself, they can actually just put these phospholipids into water, and this will self-assemble this membrane. It'll form a little bubble with an intact cell membrane around the bubble, water on the inside, water on the outside, interacting with both sides of this bilayer. So you can say that maybe this surface is the interior of the cell, which is full of water. This surface is the exterior of the cell, which is in a water medium. And so all of these polar regions are interacting with water. The water is happily interacting with them, and the hydrophobic regions are sandwiched in between these, not interacting with water at all. And we'll talk much, much more about the properties of these cell membranes when we get to cell membranes and regulation of transport across cell membranes in a couple chapters. But just keep in mind right now just how critically important this kind of structure is to the origin of life on Earth. Remember, none of these regulatory processes that create homeostasis are possible without this lipid bilayer separating the interior of the cell from the outside world. The last of the lipids I want to talk about just briefly are steroids. So steroids are a family of lipids. They're distinguished by a four-ring structure. So here we see the chemical structure of a steroid with four rings and some hydrocarbon tail attached to it. Steroids differ from each other by the functional groups attached to the carbons in the rings, so we can have different functional groups attached at different points on this ring. That's going to make these steroids have slightly different properties and serve different roles in the cell. So examples of these steroids are hormones such as estrogen and testosterone are steroids that are actually synthesized from a simpler steroid called cholesterol. Cholesterol is a really important component of plasma membranes. We'll see when we start looking at membrane structure and function more closely that cholesterol regulates how other proteins and things that are embedded in the membrane, how much they can move around in that membrane. So this is a very important structural component of cell membranes as well. So to wrap up this web lecture, I want to come back to the very, very first day of class and remind you of this list of things that are alive do. And let's think through the different biological molecules we just talked about and how they contribute to these different functions of life. So first of all, if we think about homeostasis, obviously those phospholipids are hugely important to forming that cell membrane that allows internal conditions to be maintained differently from the external conditions surrounding it and be able to maintain a stable conditions within the boundaries that are required for life. Also, steroids play a big role in, as part of the cell membrane in regulating materials entering and exiting from the cell. We saw that carbohydrates and fats are both very important for energy storage, so this ability to gather, transform, and use energy from the environment. So this idea of transmitting information through space and time. So we think partially about reproduction, but also cell signaling and communication. Steroids, remember, were important as hormones in different organisms, and so communicating between different parts of a multicellular body and signaling between cells are both things that steroids can do. And also the ability to sense and respond to the environment. So we saw that carbohydrates have a role in cell identity, so being able to detect if something in its environment is another cell from the same organism, or if it's something it needs to worry about a little bit. This is part of the cell's ability to respond to its environment. And steroids also, in their role as hormones, it's going to send signals through the body in response to inputs from the environment that coordinate bodily responses to those stimuli. So that's also going to be really important. So as we go on next time, we're going to look at nucleic acids, which are obviously going to be very important for this reproduction part of the list, and proteins, which are going to be important to many, many facets on this list. And we'll come back to this again at the end of that web lecture to summarize how these proteins and nucleic acids relate to this list of things that living things do.